introduce our next speaker. So next up to tell us more about gene therapies is Dr. Amanda Haidet-Philips. This is a packed session, as the whole day is, so I can't really tell you much about her. Again, go check out the speaker bios. But I will tell you a few things. Currently, she is the Director of Translational Medicine at Novartis Gene Therapies. And actually, they just recently changed names. So you can see the new name of the company on her slide here, which is Novartis Institute for Biomedical Research. Prior to taking on that role, she was a scientific portfolio director at the Muscular Dystrophy Association, which of course is one of the valued sponsors for today's convention. And I can tell you that she's also one of my favorite members of the CMT Research Foundation's Scientific Advisory Board. Dr. Haidet-Philips is smart, incisive, and dedicated to the cause. Dr. Haidet-Philips.
All right, well, thank you very much for the introduction. I hope today in this presentation, I can just give you a little introduction to gene therapy and convey some of the excitement as well as some of the challenges to using this approach to treat disease. So I'm a research scientist by training, and I worked for many years in the lab working to develop therapies for neuromuscular diseases. Then worked for a nonprofit organization for a while and eventually moved into biotech because I was so excited about the potential for gene therapy and I wanted to get involved there. I want to mention briefly that my talk is not going to cover any Novartis related research related to CMT, but I will talk about the gene therapy field in general and take you through some examples from other diseases and then tie that back to CMT at the end. These are my disclosures. So obviously I'm an employee of Novartis. So gene therapy is a therapeutic approach that directly targets the genetic cause of disease. It's been most widely evaluated for monogenic diseases. These are diseases that are caused by a single gene. So it could be that there's either not enough of a certain protein that's produced from a gene, or there's too much of a certain protein that's being produced from a single gene. An example of this would be like PMP22. So gene therapy introduces new genetic material, so DNA, into the target cell. And in the case of CMT, this target cell might be a cell of the nerve, either a nerve cell, a neuron, or a Schwann cell. The target cell then expresses the missing protein or is able to decrease expression of a specific protein in the case of too much protein, and this allows it to treat the disease. So the concept of gene therapy is not entirely new. It's been studied for the past 30 years, with the first therapeutic gene transfer in patients happening back in 1990. But in recent years, due to advances in the types of gene therapy vectors used, there's been increased interest, as you can see here in this graph. So the number of investigational new drug applications for gene therapy is rapidly increasing. These are what gets submitted to the FDA in the United States to allow for testing of a new gene therapy in a clinical trial. So this is a really exciting time for the field. So one of the reasons for the increased excitement and interest in gene therapy is because the field has found and tested a specific viral vector that's quite useful in gene therapy, and it's called adeno-associated virus, or AAV, and you've already heard a little bit about AAV today. So AAV is a non-pathogenic virus in humans, meaning that when we get infected just naturally, we don't get sick from the infection. The AAV that's used in gene therapy, though, is a little bit different than the one that's found in nature. So this modified AAV cannot replicate to form new AAVs, and it's not contagious to be passed to others. So AAV is currently being tested in clinical trials in a number of different neurological diseases that are shown here. And there's an AAV gene therapy that is FDA approved for the treatment of a disease called spinal muscular atrophy, or SMA. And in trials, patients with muscular dystrophy, myotubular myopathy, and SMA have shown improvements in motor function after treatment, and with improvements in breathing function and feeding also seen in some of these diseases. So I want to quickly walk you through the SMA story here, because I think it's a great success story of how therapy development can be very impactful with the right collaboration between industry, researchers, clinicians, and patient groups. So for those of you who aren't very familiar, SMA is an awful neuromuscular disease affecting mostly infants and children. The most severe form of SMA is shown here. And this is where babies present before 6 months of age, and they lack muscle tone, they have poor head control, and they never achieve the ability to sit up on their own. Prior to treatment, these type 1 SMA babies typically did not survive past their 2nd birthday. 5 years ago, there were 0 approved therapies for SMA. And today, we have 3 impactful approved therapies that all are targeting the genetic root cause of the disease. So I'm going to tell you a little bit more about the gene therapy that's approved for SMA. It's called Zolgensma, and it was developed by a company called AAV-based and then Novartis. So Zolgensma is an AAV9-based gene therapy where the AAV vector is basically a delivery vehicle for the DNA that's shown here. So part of the DNA inside the AAV is the human SMN gene, which is the missing gene in SMA. AAV9 is a special type of AAV that has the ability, when administered into the bloodstream, to cross the blood-brain barrier and get into the nervous system, particularly into the types of cells that are affected in SMA called motor neurons in the spinal cord. It's important to know that this is a one-time treatment. So patients get 1 infusion of the therapy, and then afterwards, the DNA that's delivered by the virus stays inside the cell, and it continues to produce the missing SMN protein. So I've been very fortunate to be able to be involved in some of the early development of Zolgensma, and it just continues to amaze me the impact that you can have by targeting the genetic root cause of a disease. On this slide are some of the patients that were involved in the first clinical trial testing Zolgensma. These were really brave families who agreed to try this approach and really be the first to have it be tested in their children. These children were born with SMA type 1, which I mentioned before is lethal, and really it causes children to never to be able to sit on their own. So you can see here that 11 of the 12 patients who received the therapeutic dose of Zolgensma were able to gain the ability to sit on their own. And all 12 were alive at their 2nd birthday, as compared to only 8% of children who were enrolled in a natural history study. That's a study where you follow the course of disease without treatment. And it's been so exciting to continue to hear updates as these children get older and are starting kindergarten and progressing through many other milestones that kids do. So for SMA, I really believe that there are several key factors that help contribute to the drug development success, and these are a learning for other diseases. To start, there was amazing researchers which helped uncover the genetic cause of the disease, the target cell types, and develop excellent animal models. This was all the scientific foundation for therapy development. Secondly, there were very strong SMA patient organizations that helped to increase disease awareness, disseminate information to the community, and truly enabled newborn screening in the United States. And this has led to earlier diagnosis and treatment. There were also very dedicated and engaged clinicians working together to develop robust natural history data sets and outcome measures for SMA. So these are key in showing the impact of the therapies tested in trials, since there was really clear information that had already been collected on what the disease looked like without treatment. And then lastly, there was very brave and altruistic families who participated in patient registries, in natural history studies, and in clinical trials. And some of these clinical trials even had placebo groups. These families really enabled therapies for the next generation of SMA kids. And so I think it's so important to consider, for everyone, how they might get involved in research, even if it's natural history or non-drug studies, because these are so very important to allow to lay the foundation and allow drug development to happen then in the future. So in CMT, as you've already heard, it's really an exciting time for gene therapy, and that there are approaches that also use AAV in development for many subtypes. And some of these therapies have even been licensed by biotech companies for further development. These gene therapy approaches use similar AAV vectors to what was used in the SMA example that I went through, to either deliver a missing gene or to knock down or reduce, protein from a gene that's expressing too much. It's important to mention that these approaches are all currently in preclinical development, so they have not been tested in humans in any clinical trials, but we hope that they, progress in development in the future. I think it's also, important to share that while gene therapy offers potentially transformative impact in disease, that we're still really in the early days and we're learning from our experiences. Some of the challenges that currently face the field of gene therapy are how to figure out the right dose to deliver and how to deliver that dose, what delivery route to use to make sure that you're targeting the cell types that you need for that specific disease. How do we tackle diseases with really big genes that don't fit into AAV, or diseases where you need some sort of innovative approach, or where you need a specific amount of protein expressed and not too much and not too little? How do we minimize and manage the body's immune response to AAV? And how can we deliver gene therapies to people who might have already been exposed to AAV during the course of their life? Because currently these people are excluded from gene therapy trials. And then lastly, and I think most importantly, how to deliver AAV gene therapies in a way that is very safe and ensure that we continue to learn about the potential safety risks and how to monitor and mitigate those risks. So just a few take-home messages. Gene therapy is a promising approach for the treatment of neuromuscular disease. I hope you appreciated the story of SMA. I think it's a great example of a success story for gene therapy, providing transformative impact for patients. Gene therapy, though, for CMT is still in the early stages of development, with currently being tested in animal models. So it's important to remember that challenges, still remain in the translation of gene therapies from animal studies into human trials. So this process can take some time. And especially for complex diseases like CMT, where there's, many different genetic causes, differences in symptoms between patients, different disease courses. This really, this type of disease really requires scientific innovation and strong collaboration between industry, patients, clinicians, and researchers. But, I do see so much promise in the development of therapies for CMT, between gene therapies, the antisense oligonucleotides and other approaches, especially after listening into the researcher day that happened yesterday. I think there's a really bright horizon for CMT, and I really applaud all the researchers and doctors and for the foundation for everything that they're doing. So I think that's my last slide. Thanks.
All right, thank you very much, Dr. Haidet-Philips. Really appreciate that.